Good afternoon. I'm Ellen Siegel, Chair of Friends of Cancer Research and founder. It is actually nice to be welcomed to Vivaldi's Four Seasons, and I guess it's the appropriate season, fall. We thank all of you and all of our partners who have worked so hard on this real-world evidence project. It's hard to believe that this started in 2018. Everyone has worked really hard, and you will be hearing a lot of the results. So a giant thank you for all those who participated. And also a very, very special thank you for all of those at Friends who have worked so hard, Jeff Allen and the entire team that have worked on this project. And I look forward to hearing from Jeff and look forward to hearing more about the results today on this project. So thank you and welcome.
Thank you so much, Ellen, and good afternoon, everyone. Thanks very much for joining this next installment of our real-world evidence pilot projects. As noted, this has been a great collaboration with a number of different experts and organizations that have helped design these pilot projects, and I'm honored to be able to have the chance to present some of the findings for you today. Additional information can be found on the analyses that were performed in the white papers that were distributed in advance of the meeting. In addition, following the initial presentation of results, we'll hear from a number of the experts that helped design this and participated in the analyses to provide additional detail and insights into Pilot 2.0. But first, let's take a few minutes to begin to review some of the initial findings that were achieved by our collaborative data partners. Today's data presentation and their discussions following will be representative of the assessment of real-world endpoints across treatment groups. We've called this the results of the analysis using Pilot 2.0 data, with 10 of our data partners who have performed these analyses for today's discussion. For sake of background and a little bit more information on the initial proof of concept Pilot 1.0, for this project, 6 of our data partners initially focused on evaluating the performance of real-world endpoints across multiple data sets by focusing on a common question. And that question was, what outcomes can be evaluated for advanced non-small cell lung cancer patients treated with immune checkpoint inhibitors? Through these findings, we characterized the demographic and clinical characteristics of advanced non-small cell lung cancer patients that were treated with PD-1 or PD-L1 inhibitors. We also assessed the ability to generate real-world endpoints, including real-world overall survival, real-world progression-free survival, real-world time to next treatment, and real-world time to treatment discontinuation. What we were able to see from this initial pilot project was a large number of common characteristics between the data sets, despite the inherent differences between the sources of data and the processes used to capture it. So this confirmed that the feasibility of being able to construct these data sets. One observed difference, however, was that the differences of available death data that was contained in each data source was variable. And we'll hear more about that in some of the subsequent presentations later today. In addition, Pilot 1.0 groups were able to demonstrate that several of the real-world endpoints correlated with overall survival. In addition, the overall survival observed fell within the range of median overall survival that has been observed in several clinical trials. Of course, further evaluation of these real-world endpoints is needed in order to demonstrate the extent to which they could be potential proxies for overall survival. The performance of Pilot 1.0 left several questions remaining. One, can real-world endpoints be used to accurately characterize differences between available interventions? And can further alignment based on data quality standards be used to develop an analytical framework to evaluate real-world endpoints? And we'll begin to explore those through Pilot 2.0 in our discussions today. During today's meeting, you'll hear how several other groups have applied the approach that was developed as part of Project 1.0. The first was using the cancer analysis system in the UK, which has a definitive date of death data available with it. The second has applied this framework created for Pilot 1.0 in the disease setting of melanoma to better understand whether these findings extended beyond the advanced non-small cell lung cancer. But first, we'll discuss the Pilot 2.0 and two specific work streams that will be described through this project, including a treatment comparison analysis that we'll discuss today, as well as an ongoing work around internal consistency analyses. To begin with Project 2.0, again, the focus of this was to explore the potential endpoints that may be fit for assessing outcomes associated with a product compared to an existing alternative. In this project, we wanted to revisit those endpoints that were obtained in Pilot 1.0, including real-world overall survival, time to treatment discontinuation, and time to next treatment, to better understand how these could be used across two frontline treatment pairs in advanced non-small cell lung cancer. The first objective of this project was to describe the demographic and clinical characteristics of patients with non-small cell lung cancer receiving frontline doublet chemotherapy, PD-L1 monotherapy, or PD-L1 plus doublet chemotherapy in combination. The second objective was to compare the real-world outcomes among the selected frontline therapy pairs in order to explore the extent to which these different measures could be used to distinguish between the treatment options. I want to recognize the various data partners that have contributed their time, their expertise, and their data in performing these analyses, including ASCO CancerLink with Concerto AI, CODA, Flatiron Health, IQVIA, Kaiser Permanente with the Cancer Research Network, the Mayo Clinic with Optum Labs, McKesson, NCI SEER, SciEps, and Tempus. Our 10 partners have put in a lot of time and effort and able to make this project successful. We very much thank them for their contribution to this ongoing body of work. The definitions that were used in order to assess the real-world performance of these different therapeutic interventions are listed here with the different definitions that were applied. I won't read them verbatim for you. These are all contained in the white papers that summarize the results of today's discussion, but these have been applied consistently across each of the different data sets and carefully defined by each of our participants. I should note that with regards to time to next treatment and time to treatment discontinuation, these include death as an event, whereas in Pilot 1.0, these definitions did not. So in order to move into some of the results that were observed, this slide depicts the percentage of overall survival at 12 months by treatment type. As you can see, in looking at the observations from each of the different groups denoted by letters at the bottom axis, there is a proportional improvement that is suggested among several of the data sets when looking across the different treatment options. It is worth noting that all the data that will be described in this presentation today is unadjusted and conducted on a diverse patient population without further restrictions. Of interesting note, when looking at overall survival, in other objective endpoints, these are susceptible to factors such as post-baseline events, like subsequent therapies. It may make the treatment effects harder to interpret. It may also suffer from substantial missing mortality data, incomplete follow-up time, and patient crossover. These are all reasons that could contribute to variation between the different data sets and make overall survival a challenging endpoint at times to definitively assess between these different treatment options. But nevertheless, what can be seen here is the directionality of an IO treatment effect was demonstrated in the real-world results. While there's a wide range of estimates across data sets, those with IO included tended to have a higher 12-month overall survival than chemotherapy-treated patients. Though you can note that there is overlap between the confidence intervals. On the far left-hand side of the graph, you can see the SEER data set, which is identified and provides a population-based reference. But it should also be noted that the SEER data is based on Medicare, so it has no younger patients, whereas several of the participating groups had over 1/3 of their patient populations younger than those that might be captured within the SEER data. Other population differences weren't controlled for as well, such as differences of staging that might be seen within the population. On the right-hand side, it's worth noting that the smallest number of patients are in the IO doublet group, and this contributed to the broader confidence intervals that you can see around each data set. This, in large part, is due to the recency of time that IO and doublet therapy was adopted into routine clinical care. As a measurement of time to next treatment, this slide depicts the fraction of patients who have not started a next treatment within 12 months. Here you can see that time to next treatment shows a more definitive trend toward increasing for IO-containing treatments. This may be an indicator that patients are staying on their treatment longer because their cancer has not progressed. So it should be noted that time to next treatment is a challenge as an endpoint if a treatment is on a fixed number of cycles. Again, on the left, you can see that the SEER data set is noted as a benchmark. And it should be noted that on the right-hand side, the Group C information was dropped due to incomplete follow-up time for the doublet treatment regimen. But similarly to what was observed with time to next treatment, this slide depicts the measure of time to treatment discontinuation by demonstrating the fraction of patients who have not discontinued treatment at 12 months. As a reminder, time to treatment discontinuation was defined as having a subsequent therapy, 180 days without therapy, or a date of death while on a frontline therapy. A similar trend toward time to treatment discontinuation extending for patients who were treated with IO-containing regimens was observed. Some of the variation that's seen between the different data sets may occur based on the source of the data. For example, with claims data, the data may be more longitudinal, which could take more time in order to show a discontinuation event, where EHR-based data could be more rapid. So in conclusion, for the summary of these initial findings from Pilot 2.0, we saw that it is possible to coordinate the efforts across numerous real-world oncology data organizations to reach a high level of alignment on important data elements and definitions for real-world endpoints in the context of a focused research question. The depth of data varied across the data providers, and distinct characteristics were identified among the cohorts provided by each organization, likely attributable to the characteristics of the data source and the underlying population it's capturing. We saw that it's possible to identify a similar directionality in treatment effect of IO as compared to chemotherapy in real-world evidence, which is consistent with findings in recent clinical trials. And finally, we demonstrate the correlation between real-world overall survival and the other real-world endpoints, including in data sets with near-complete mortality data, which indicated the potential use of real-world time to treatment discontinuation or time to next treatment as a proxy endpoint for the treatment effectiveness in real-world studies. I want to thank each of the data organizations again that contributed to this effort, along with our key collaborators from the FDA and the NCI and the other advisors who have helped make this work possible. We have an exciting meeting today, but first I'd like to turn to a panel of our experts who will further discuss these results and the observations that they were able to obtain through their work with this data. And I'd like to turn things over to Joe Henk from Optum Labs. Thank you. Joe.
Thanks, Jeff. I'm, as always, delighted to be here. So I'd like to welcome my fellow panelists, Dr. Elizabeth Garrett-Meyer, who's with the Center for Research and Analytics at the American Society of Clinical Oncology, Dr. Sean Cozen, Janssen Research and Development, Dr. Donna Rivera with the FDA's Oncology Center for Excellence, and last but not least, Dr. Nick Robert from McKesson Life Sciences. Welcome, everybody.
Welcome.
Thank you.
So a little bit of housekeeping first. If you go to the bottom of your screen, attendees, there is a question and answer function. If you wish to ask questions, we will see them, the other attendees will not. And if time allows, we'll certainly try to get to them. So the title for this panel is Methods for Optimizing Real-World Data and Endpoints. And this whole effort, as Jeff has said, has really been a collaborative effort by Friends of Cancer Research and many collaborating organizations, and it's been a pragmatic effort in the sense that we're actually touching data. And the title really says it all, doesn't it? Optimize, to make the best and most effective use of data and endpoints. So what I'd like to cover in our short time with our panelists today is discuss with them how they see us being able to optimize the use of this real world data and to develop or produce real world endpoints. So jumping right on in, Sean, I'm going to ask you the first question. What does optimizing real world data and endpoints mean to you? What opportunities lie ahead? What data can we best optimize the use of?
Sure, thank you, Joe. It's great to be here. That's a great question. There are different ways of thinking about that. One of the ways would be to think about optimization as a function of three different components and along three axes, if you will. And those three different components are technology, data, and also analytics. And in recent years, we've made progress along these three axes. So in terms of what we can optimize, it's all dependent on these three dimensions in a way, which are not mutually exclusive. They in fact influence each other. For example, in terms of technology now, we have new opportunities to collect deeper data assets in the real world. For example, instead of relying solely on the radiology report to extract tumor response and tumor dynamics using natural language processing and human adjudication, in some cases, we now have access to the actual DICOM images, so deeper data assets that can potentially allow us to optimize how we calculate real-world endpoints. And in terms of, so that's technology allowing us to collect deeper data in the real world. In terms of analytics, the analytics that are involved when we can potentially access the actual images, the actual DICOM images versus the radiology reports are somewhat different than the types of analytics that we're now relying on to calculate tumor dynamics. So optimization in that scenario gives us the opportunity to apply more advanced analytical methods, potentially, for example, image recognition, AI, and ML, in understanding tumor dynamics beyond what may be accessible through the radiology reports. So that's one way that one can think about optimization along these three non-mutually exclusive dimensions or axes.
Thank you. Nick, so Sean started to introduce some very exciting and perhaps at this point aspirational, but in looking at what Jeff presented, a question that I have is, how did you settle on overall survival, time to discontinuation, and time to next treatment instead of some of the other metrics?
Well, so what's exciting about our work is that we're taking real-world evidence, real-world data, and trying to translate it and look and compare this with outcomes from clinical trials. So the path moving forward in medicine and in oncology has really been the tried and true gold standard clinical trials. But I think many of us in the real-world data area feel that there's certainly an opportunity to leverage all the data we have on patients, often treated with similar regimens, certainly after a regimen is approved. So I think there are lessons learned from those that do clinical trials to our work. And I think part of it is not being too ambitious and being careful to define our endpoints. If you look at clinical trials, for example, there's a lesson about how they address the issue of response. People sat down and formed a consensus and identified an endpoint response in a way that was reproducible and you could compare trial to trial. So we have a similar challenge in looking at real world data. We have strengths and weaknesses, and I think we really need to be in a situation that we can work within our boundaries. Response, progression-free survival, we'll hear more about that, is challenging when you look at real world data. On the other hand, time to treatment discontinuation, time to next treatment, and overall survival, though challenging, are much more achievable with real world data. And we'll speak maybe more later about this, but there's a difference between looking at structured data and unstructured data that can give us more or less information about these three endpoints.
So piggybacking on that, the one data component endpoint that was not looked at or presented was progression. Can you elaborate on what you just said and comment on the endpoint progression, time to progression, and where you see it fits in real world evidence?
So I think if you're dealing with structured data, it's, at least with the McKesson data, I would say it would be very difficult to establish progression. We can talk about time to next treatment, but to actually, without opening the charts, really assess progression. When we do open the charts, we certainly look at progress notes, and we use the physician assessment, which is usually based on radiological reports. And we've, within our group, developed guidelines for those definitions. Again, one needs to be cautious, because if you were doing a clinical trial using RECIST data, that's much more firm. Remembering that in a clinical trial, you are prospectively decided when to repeat tests. In the real world data situation, this is not done in a systematic way. Tests are done and often repeated, but not necessarily every 3 cycles like in a clinical trial. So again, I think the challenge is for us to be transparent and identify what we can do and what we can't do, and then share that with those that review our literature.
Thanks, Nick. Hey, Donna, nice to see you again. So endpoints are really dependent upon the definition, and this is where we're getting into with Nick. The definition and easy for me to say, operationalizing the outcomes like a metric, overall survival, but then also on the statistical estimation of the effect, like a hazard ratio. But thinking of the former and your involvement in these projects, and the fact that these projects did not aggregate data sets, they harmonized a process or a methodology. Can you share some of the challenges that this entailed to, so to speak, harmonize operations between all 10 groups?
Sure, thanks, Joe. So I think one of the things is really how to agree across so many of these different heterogeneous data types, and really thinking through how we define these items up front. I mean, I think when we look at the endpoints, and something I think all of us really have tried to understand is comparability. So much of this is thinking about the context of trials and real world data. And I think it's important to recognize that this is really not an either or type of conversation, but really where real world data has this opportunity to provide complementary information to the gold standard trials and really expand in areas such as rare populations where maybe there is not sufficient knowledge already. So I think when we're thinking about definitions and we see these comparisons, real world data often shows that patients are doing worse, but that is largely translational to the fact that there's strict RCT criteria, right? Inclusion, exclusion, what Nick alluded to, definitions of events. And so I think comparison, when we think about defining these endpoints, you aren't really comparing the same population, right? You have a strictly controlled population versus a real world population. So we're not necessarily expecting the same outcome, and we're not expecting the point estimate necessarily to be replicated. So I think initially that's a challenge. And then another challenge for us was defining upfront, ascertaining upfront what's going to be the most logical way to compare. And how do you validate something in comparison to a trial, knowing, as Jeff mentioned in the presentation, the differences in the underlying population. For example, when we think about the internal consistency study, which is where we looked at Keynote 189 and then the characteristics of these data, can we replicate the inclusion-exclusion criteria and see how these different data aspects sequentially affect outcomes? And what about, you know, hepatic function or PD-L1 data? What's the granularity of data required? How does the granularity of data required translate to the outcomes, translate to the definition, for example, of time to treatment discontinuation, right? And in claims data versus EHR, how are we measuring those? How are we measuring enrollment? And really, how are we understanding all of these in the context of statistical methods? Because we know the temporality of these aspects is different.
Thank you. Liz, you're not going to get away with no questions. So thinking about it, there's certainly challenges in operationalizing the variables and standardizing the data sets that we use as we try to be have as much commonality as possible. But in the end, we know that the analyst and the statistician have to clean up all the mess. So Liz, what are some of the challenges? You've been involved in this pilot 1 and 2. What are some of the challenges we've faced, and what can be done? What can be adjusted for in the analytic phase? And then, as importantly, what can't be?
Yeah, so thanks, Joe. That's a great question. So one of the things that I think that we did do that was really helpful is what we should always do, which is to hold ourselves to the same standard as we would with human subjects research studies, whether it be a clinical trial or an observational study or a real-world data set that has de-identified data. Just because these data are de-identified and might not have to be, the project might not have to be reviewed by an IRB or a scientific review committee, that doesn't mean that we shouldn't have a very rigorous, well-defined plan in advance that makes it very clear to everyone on the project team exactly what is going to be done within the project. That's critically important for the statisticians to have well-defined objectives, endpoints well-defined, and an analysis plan laid out well in advance so it's entirely clear exactly what the expectations are. It also fosters reproducibility. So it's really important after the study is done that we can say to others, this is exactly how we analyze the data. Now, in our situation with this collaboration across these different groups, we knew from the beginning that if we didn't spell everything out very clearly in the beginning, we were going to get to the end and we were going to find things that might even have been tiny differences could have blown up into, oh, well, none of our estimates are comparable because this group did this thing slightly differently than this group, et cetera. So I think that's one of the things that we did quite well. And we know, all of us on the phone, that as we went through the process, we thought we were entirely clear in the very beginning, and we still found that there were some things that were not as well defined as they need to be, that we needed to go back and provide a little bit more detail. In addition, just thinking in advance about things like prognostic factors when we're dealing with randomized studies. We obviously care about prognostic factors, but we're not as focused on including them in the analyses in most cases due to the randomization. So that's something that we need to think really clearly about, how to incorporate them, what things we need to pull out from the data in advance. In these kinds of studies, when the data are cut, you don't have the ability necessarily to go back and get more data. So you have to think in advance about what variables you need before you start the data analysis. And then the other thing, I think, as much as I say that we need to have a really strong plan in advance and think it through clearly, we also have to be open to some additional analyses when things either don't go exactly as planned or things come up that we just really weren't anticipating. So, you have to be transparent about these things. But for example, we did a sensitivity analysis in this project. Once we realized that a follow-up on the patients in the chemo doublet arm was substantially longer than in the immune checkpoint inhibitor arm. And we were concerned that that might have an impact on the estimated hazard ratios. So across the groups, we said, hey, let's do another analysis where we censor all of our endpoints for overall survival at 24 months. And then let's look and see if our results are consistent or not. So that was a sensitivity analysis that we thought through and did really after the fact. But we wanted to, ensure that the assumptions that we went in with were met, and so we added that on after the fact. to try to address something that came up that we really hadn't thought about in advance.
So one question I have methodologically is crossover happens theoretically in design in a clinical trial. And in real life, operating in, I would say, in an economy when everything else is going on, we have constraints. It happens. Now, in our project that Jeff just highlighted, we primarily looked at frontline. So I would define crossover as those who go to second line, for instance. Is this a bigger issue with observational data than it is with trials, or is it the same issue with perhaps the same solution analytically?
Yeah, well, so it has become more of an issue in trials. I mean, we're seeing more and more trials that are considered definitive phase 3 studies where progression-free survival is the endpoint because of this issue of crossover. So it's not something that trials are immune to. You know, when I think about it from the real world setting, you know, this is about as real world as it gets. When you assign somebody to a treatment and then they, for whatever reason they stop that treatment, they're going to get whatever the next line is, whatever they and their physician decides is the best thing to get. So it really helps reflect what the real world experience of patients might be. And sometimes, you know, what it ends up being is a sequencing question, right? What do you get first versus what do you get second? And in some cases, it might not necessarily matter. So the real world setting, what it really provides us with is more of a question of the effectiveness of being prescribed that medication first versus potentially second. Whereas the trials are really more focused on a question of efficacy in most cases. If the patient receives this under these conditions that are pre-specified according to this protocol, how effective is the treatment? So the real world trials give us, you know, a slightly or maybe more dramatically different answer. But that's why I think what we see in these studies is we see probably effect sizes that are not as dramatic as we see in the trials because they're allowing for all of the heterogeneity that happens to people outside of the trial setting.
So Donna, going back to where I started with the identifying the variables, and what I omitted to mention is identifying variables that will help us identify the population. Do you see synergies with what Liz is you know, stuck with handling on the back end, so to speak, is how I think about it? And what can we do to help our statistician friends?
Yeah, absolutely. I think design is something that is really important, right? So what we can ascertain up front, what we can be rigorous in designing our cohort and really thinking through what is the question, what is the data available, and what is the purpose, this kind of fitness for purpose that we refer to, and really, really thinking through, can you answer the question you have with the data that exists? Can you match on baseline characteristics? And what's the granularity of the data required, right? Is a binary variable, yes, no, acceptable as a proxy? Do we need lab values? Do we need continuous data? Is categorical, you know, variable acceptable? And what is the discrete level we really need to draw to the outcome? So I think really thinking carefully about who is in the cohort and really understanding that is imperative in this conversation and helping on the design front to then avoid some of the things that you would have to do on the analytics side. And I think it also gets to the question of external validity, because very much the truth that we all are seeking as to, you know, is this medication, you know, really related to the outcome and isolation of the treatment effect. I think it really depends on who you're talking about, in that, you know, who is being studied is really directly correlated to who the evidence pertains to. So the population that's being studied is exactly who you would need to understand what evidence you're generating and who that evidence is related to. So I think it's really that question of also external validity. And we'll probably get into internal validity a little later.
Joe, can I make a comment?
Please.
So I think just following up on Liz's comment, one of the things that is a little subtle, but when we looked at our data, people received chemotherapy for metastatic non-small cell lung cancer. And then once IO therapies became available, people stopped getting chemotherapy. So when you look at our data, there are snapshots in time. We're not doing what's in a randomized trial. We're not randomizing patients to chemotherapy versus chemotherapy plus IO. We're actually looking at patients who got treated with chemotherapy. And then once IOs became approved, we look at how those patients. So the crossover issue, it's a different challenge. It's not like in a clinical trial where you can potentially cross over. So I think that's something we have to remind ourselves. And I think it's important to be transparent. That's how we collect our data. In terms of Donna's remark, I think it's also important, and to emphasize what she has said, something as simple as distinguishing de novo metastatic cancer, someone presenting with stage 4 disease versus someone who presents with a history of lung cancer now with metastatic disease. You think that's easy to extract, but if it isn't extracted properly, you're really going to see two different outcomes. So it is really important, as it has been said by others, to sit down very carefully and plan your endpoints and plan your criteria, because if it's not done right at the beginning, it leads to, as you heard, to a lot of potential confusion.
Thank you. I was going to ask if, Sean, I'm going to shift gears a little second. I see that we're running a little short on time, and I'm going to move on to next steps as far as the panel and the efforts. And Sean, one question for you is that the outcomes studied here, other than overall survival, the time to discontinuation and time to next treatment, aren't typical outcomes from clinical trials. Do you see any potential to use them or include them as part of clinical trials going forward?
Sure. But these are endpoints that we have used in the past in clinical trials. But you're absolutely right. Because we can precisely, as precise as things can be in traditional clinical trials, measure progression-free survival and tumor progression response. We start to abandon actually time to treatment discontinuation and time to next treatment, which were endpoints that did emerge from the world of clinical investigations. So I think it's important for us to keep that in mind and to, in fact, look at these endpoints in traditional clinical trials so that we can have a benchmark in terms of the modified endpoints that we're driving out of real world data. Because benchmarking what we do and how we do things in traditional clinical trials is critical to optimizing how we use and how we analyze real world data. Because when we say optimization, that means we have a gold standard in mind. And if that gold standard is traditional randomized clinical trials, it's important to establish where the bar when it comes to data integrity, when it comes to endpoints, for example, time to treatment, discontinuation, and next treatment, where that bar is in traditional clinical trials. And in fact, some of those standards and parameters haven't yet been defined. So there's an opportunity right now to look at how we do things in clinical trials, try to establish quantitative benchmarks that we can then carry over as we optimize the use of real-world data.
Thank you. So I'm going to open this up a little bit. We're going to try doing this on Zoom. I'll just ask that you raise your hand so I can see you. I want to ask the same question to everybody, and that's basically what next? And when you answer that question, I'm really interested in other endpoints, perhaps adverse effects or toxicities. And we've heard people speak about this in the past, that's certainly a current use of safety studies, et cetera, that do use real world data. But who would like to go first? Okay, Donna will go first. You raised her hand, I swore I saw it.
Sure, I'll go. So, I think, Joe, you just alluded to this, right? Observational research has a longstanding history of evaluating adverse events, right, in pharmacoepidemiology in this space. And so I think the question is the translational applicability of real world data across, the continuum of potential outcomes. And I think one of the key points to this, as far as next steps from my perspective, is really expanding on and highlighting the importance of data quality and optimizing the data quality. I mean, I think there's a critical unmet need and having a consensus on how we look at real world data quality and how we truly assess it. And any of the endpoints that we would develop or any of the new methodologies really rest on that data aspect, right? Because, you know, clinical trials are the gold standard, especially for efficacy. However, we understand there's limitations and data is imperfect, and real world data is in a less controlled environment with a great room for selection bias, residual confounding, immortal time bias. So with these, it's really, I think, emphasizing the need for data quality standards and that internal validity component, right? Of if you have strong data that's very well, you know, aggregated and understood going in, then the ability to measure additional outcomes is going to be, you know, much more rigorous.
Liz, what would be your, what's getting you excited? What do you want to see next? Maybe pilot 3.0 or 2.2? Yeah.
Well, I mean, what I was thinking of in terms of response to your question was, so we're familiar with when a clinical trial is submitted to a high-tier journal and is published, the protocol is made available along with the results to the trial. So there's the manuscript and there's also the protocol. I really feel like in the interest of some of the things that Donna said and in the interest of reproducibility, we should be making available not just the results, but the methods that go into the results. You know, when I first started working with real-world data, I had a long experience working with clinical trial data and observational studies, more traditional prospective observational studies. And I really had no idea how different this space was going to be in terms of issues of measurement error, and when I say measurement error, systematic measurement error, in addition to tons of missing data. And they just happen in a way that they don't in prospective studies. So I think that we really have an obligation to not just allow these studies to happen without really clearly defined explanations of how they were done, but we really need to make that information available and hold people to certain standards for real-world data analysis. So maybe Pilot 3.0 would be coming up with standards for reporting in a way that we haven't really laid them out before.
Nick, what's your perspective on the?
Well, I think.
What are you doing next?
Yeah, the potential is exciting. We've talked about the potential of extrapolating outcomes of a clinical trial to patients that wouldn't have been eligible but still derive benefit, expanding indications, you know, treating patients that may have not been in the trial but have been treated in the real world. But I think an area that's very exciting, very challenging, it's not there yet, but we are appreciating that there are uncommon cancers that it's very hard to do a randomized trial, like, you know, certain skin cancers, for example. But there are also, as we dive deeper with precision medicine to divide up even common cancers like breast cancer or lung cancer, we're dealing with small subsets. Doing a randomized phase 3 trial, the time, expense is formidable. And if we, and again, I don't think we're there yet, but if we had the confidence that we could look at real world data and develop synthetic control arms, so we wouldn't have to do a randomized trial. It's going to have to be robust. I think we're going to have to unleash what Sean talked about, the technology and the analytics and the data. But I think that would be fabulous if we could achieve that, because we could get treatments out sooner and not go through the laborious phase 3 process.
And Sean, take us home. This is the last minute.
You know, I think everyone has highlighted all the key points in terms of, you know, what to do next. So I think obviously thinking about ways to define data quality in real world data assets is very, very important. And perhaps we can think about how we define data quality and data integrity in traditional clinical trials as a benchmark, which is done procedurally. So versus mathematically, the way we assess data integrity and data quality in clinical trials is through site inspections and adherence to good clinical practice guidelines. So there are procedures that are in place. So perhaps we can think about similar procedures and standards, if you will, procedural standards that we can follow to ensure data integrity in real-world data assets. And this was also mentioned by Donna, that the design of these studies are are critical. And what we can do today in terms of a study design may be different than what we can do in a couple of years because of the three sort of dimensions of real-world data that are continually improving. So thinking about the design of these studies in a dynamic fashion so that we can take advantage of technological advances is critical. Lastly, I think the more that we can share experiences and data to the extent that's possible, I think it would be better for the entire ecosystem overall and ultimately for patients.
So thank you, panelists. Thank you, friends, as always. I'll point people to the white paper, the recent white paper from Friends, for a lot more lessons learned and publications to come. I think back to you, Jeff. Thank you.
Thank you.
Thank you.
Great, thanks very much, Joe. And thanks to all of our panelists and to all the organizations and experts that helped develop these analyses of real world endpoints. I also want to highlight the white paper that Joe just mentioned. All of you should have received that in the emails leading up today to the event today. And there is a lot more information, additional analyses than what were presented and described as we move forward toward publishing those from the results of today's presentations. Moving on into this next session of results, we have presentations from two of our additional collaborators. As mentioned in the earlier discussions, one challenge to utilizing real-world endpoints, particularly overall survival, is the absence of complete date of death data. Our collaborators from IQVIA have reconstructed the Pilot 1.0 approach utilizing the cancer analysis system from the UK, which has complete death information included. So we'll be able to see how that compares. In addition, the research team from Oken extended the analyses into a data set of melanoma patients treated with IO therapy. So we'll be able to see if the initial findings regarding endpoints in lung cancer are reproducible in another tumor type. So to provide a summary of those results, I'd like to introduce our next presenters, Adam Wright, Principal of Real World Solutions for the UK and Ireland of IQVIA, Maxime Mehi, the data scientist and team lead at Oken, and Brigitte Durano, the head of the Department of Dermatology, the University Hospital of Nantes in France. Adam?
Hello, my name is Adam Reich. I'm a principal in IQVIA's Real World Solutions team based out of London. In this presentation, I will discuss the results of the CAS pilot, which sought to apply the framework established in the FRIENDS 1.0 pilot study to a different nationally sourced cancer registry from the United Kingdom. The intent was to assess the consistency of real-world outcomes such as overall survival and to further validate the previously observed observations between overall survival and potential surrogate endpoints in real-world data. The CAS pilot followed the FRIENDS 1.0 pilot study protocol, which had three main objectives. Objective one was to describe the demographic and clinical characteristics of advanced non-small cell lung cancer patients treated with immune checkpoint inhibitors, also known as PD-L1 inhibitors. The second objective was to assess real-world endpoints in these same patients. Objective three was to assess the performance of real-world endpoints such as time to next treatment and time to treatment discontinuation as surrogate endpoints for overall survival. And our CAS study had a fourth objective, which was to highlight the performance and consistency of real-world endpoints in the CAS dataset versus the 6 US datasets assessed in the original 1.0 pilot. This allowed us to see how well the work in the US translated to another geography in a different type of data source, namely a population-level nationally sourced registry versus the electronic health record datasets and claims datasets that were utilized in the initial pilot. And perhaps more importantly, we were able to apply the same study protocol to a data source which has complete mortality information, a common limitation among the US data sources evaluated in the initial pilot project. But before we get into any of the results, I think it's important to give an explanation on the cancer analysis system and describe what it is. So CAS is a cancer registry that includes information on more than 99% of all cancer patients in England. It contains data on patient and tumor characteristics, treatments, hospitalizations, and more importantly, mortality. Through linkage with the UK's Office for National Statistics, CAS provides vital status information on all patients, including death records and death date, and at certain points in follow-up confirms that patients are actually alive, which was critical in reliably assessing overall survival and validating derived endpoints such as time to next treatment and time to treatment discontinuation, which were likewise extracted in the initial pilot project. We had to adapt the framework or protocol from the initial 1.0 pilot to be able to conduct the same analysis on the cancer analysis system. So while many of the US data sources had to also make slight protocol adaptations, we had to make adaptations as well. This included slight adjustments to the study time period based on the data that was available, the patient selection, and some of the ways we conducted the statistical analysis. For example, because disease progression is not captured in the CAS dataset, we were only able to include incident stage 3B and stage 4 non-small cell lung cancer patients in our cohort. In our study, we identified 2,035 advanced non-small cell lung cancer patients treated with immune checkpoint inhibitors. Now you'll notice that our initial starting point for identifying these patients was the more than 230,000 patients diagnosed with lung cancer in England. Once you applied the time period of 2013 to 2017 and a number of inclusion and exclusion criteria, some of which were specific to the cancer analysis system, we end up with a final patient population of just over 2,000 patients, which is actually less than 1% of the total patients in our cohort. Something that is a challenge in real world data, applying inclusion and exclusion criteria that gets you at a defined group of patients without sacrificing patient volumes or statistical significance. Our final cohort size of just over 2,000 patients was comparable to the larger US data sources from the initial pilot, was representative of UK patients overall, and each patient in our data source and in our final cohort had complete records of vital status and death information. Looking at objective 1, where we want to describe the demographic and clinical characteristics, we noticed that the patient population in our CAS study cohort was very similar to the US datasets from the initial 1.0 pilot. For example, the median age at advanced non-small cell lung cancer diagnosis was 67 years in the CAS dataset, whereas the US range was 64 to 70 years. 54% of the CAS cohort was male, where the US range was 51 to 56%. And even though we weren't able to include patients diagnosed at early stages of lung cancer, 82% of patients were diagnosed at stage 4, which was well within the US range, which was actually quite broad at 62 to 91% across the various sources. And lastly, 71% of our CAS cohort had confirmed non-squamous histology, right in the middle of the US range of 66 to 74%. So this demonstrated that we actually had a very similar, from a demographic and clinical characteristic standpoint, population as what was assessed in the initial pilot presentation. Before we get into objective 2, it's important to understand the differences in PD-L1 usage between the UK and the US, as that will have a significant impact on what these endpoints look like. Starting with the fact that the US approval of PD-L1 inhibitors in non-small cell lung cancer occurred roughly 2 years before approval in the UK. And UK approval initially came in the second line, but quickly thereafter, it was pembrolizumab was approved conditionally in the first line setting. And as a result, we see a much higher proportion of PD-L1 inhibitors used in the first line at 47%, compared to the US data sources, which were at 18 to 30%. 94% of our patients treated with PD-L1 inhibitors received no subsequent lines of therapy, which was extremely high compared to the US range, which was 67 to 71%. Time from advanced non-small cell lung cancer diagnosis to PD-L1 treatment initiation was shorter at just under 5 months than in the US data sets at 6 to 8 months, again, likely due to the higher first-line usage. So understanding that there are differences in PD-L1 usage, when you look at real-world overall survival, you still see consistency in these endpoints. For example, the real-world overall survival in the CAS dataset was 11.4 months, whereas the US data sources were 8.6 to 13.5 months. So CAS was very consistent with those data sources, despite these differences, and despite the fact that there were different captures of mortality information in the CAS dataset versus the other data sets, which will lend itself to the discussion over the surrogate endpoints later in this presentation. Real-world time to treatment discontinuation was just under 5 months, which was toward the top end of the US range, but understandably at the top end because of the earlier use of these products in first-line therapy. And you would expect patients who are being treated to either progression or to toxicity to be on treatment longer if they initiate in first line. This slide is a graphic representation of the prior slide. And if you look at the CAS data set on the far right, you see that it is pretty consistent with the 6 different data sources assessed in the initial pilot, but that it also falls very squarely within the range of similar survival outputs from clinical trials, which included immune checkpoint inhibitors. So not only is CAS consistent with other real-world data sources, showing that applying the framework across a variety of data sources will yield consistent results, but that also it will yield results that are in alignment with clinical trial results. Therefore, showing that once a clinical trial has potentially ended, we could be able to look at long-term survival in real-world data sources going forward. Correlations in real-world overall survival and the surrogate endpoints of time to next treatment and time to treatment discontinuation were comparable to US data sources. And like the US data sources, time to treatment discontinuation was by far a stronger surrogate. Now, this is not surprising. When we assessed real-world time to treatment discontinuation, we would include patients who either died or who stopped receiving treatment. Whereas in time to next treatment, only patients who moved on to a subsequent treatment were included. So any patients who died were censored and not included in that analysis. So as you would expect from a data source like CAS, where complete mortality information is available, the correlation between real-world time to treatment discontinuation and real-world overall survival will be better. Plus, it's more statistically significant in that 900 patients qualified for that analysis versus the 100 patients in the time to next treatment analysis. So it's very important to consider all of these different elements when deciding which surrogate endpoints might be most appropriate in evaluating real-world data sources. And you can see by this chart that the consistency lies with the time to treatment discontinuation. So there were some very important conclusions that we were able to draw from this study. The first of which is we had established a common protocol to identify non-small cell lung cancer patients treated with checkpoint inhibitors from a variety of US data sources, and we were able to successfully adapt that to the CAS data set, despite a different population, a different mode of recording of this information, and different data access processes. Despite differences in patient geography, cohort size, the demographic and clinical characteristics were strikingly similar between the CAS cohort and the US data sets. Overall survival among the CAS cohort was very similar to the median overall survival values observed in the US data sets, and as mentioned, fell squarely within the range of what was observed in PD-L1 trials. Again, showing consistency among different real-world data sources and giving more credence to the use of real-world data sources outside of the clinical trial setting to better understand clinical effectiveness of therapies. And then lastly, extracted endpoints such as time to next treatment and time to treatment discontinuation from CAS demonstrated reasonable correlation with OS. So having complete mortality information did not necessarily result in stronger relationships between endpoints, but likely provided us with a more reliable comparison. Knowing that mortality information is fragmented in US data sources and often has to be triangulated from third-party sources, we didn't know how strong the time to treatment discontinuation information in the US data sources actually was. So being able to provide that same information from the CAS data set not only shows us that real-world surrogate endpoints can be reliable, but that what we did in the United States is potentially more reliable than what was initially expected. And then due to limited follow-up and relatively immature data, we would recommend assessing these results again with future refreshes of this information to further validate whether these endpoints are acceptable surrogates for overall survival or not.
Cutaneous melanoma is an aggressive skin tumor, and until 2011, the alone treatment was chemotherapy with only 10% of 5-year survivors. Now we have new treatment with targeted drugs, anti-BRAF and anti-MEK, and checkpoint inhibitor. With this new drug, dramatically has been improved the outcome perspective for our melanoma patients. Until now, no multicentric melanomas today Comparing between clinical trial and real world data has been performed. And for us clinicians, the real world evidence to provide long-term, robust and confirmed efficacy evaluation of checkpoint inhibitor is very important. So now we're going to focus on checkpoint inhibitors.
So in order to do so, we're going to perform a real-world data analysis. Real-world data is posed to bring a lot of value, but it has also tremendous challenges. So that's why we're doing another instance of Pilot 1.0 study for the Friends of Cancer Research Framework advancement. In this framework, we have on one side data elements that are defined and standardized in order to enable a harmonized characterization of populations in order to compare them and potentially find confounders and for other uses as well. On the other side, we have definitions for real-world endpoints themselves. There were two other instances of Pilot 1 conducted, one with 6 data partners in the US and the other one in the UK, both focusing on non-small cell lung cancer with immunotherapy. Our pilot study is going to extend those studies with another study with melanoma patients in France. So another cancer in another country.
So this work has been performed with RICMEL cohort. What is RICMEL cohort? It's a national network for research and clinical investigation on melanoma, open since 2012. Now in this database, we have 27,000 patients. The inclusion criteria are cutaneous and ocular melanoma. Mandatory data set are primitive tumor diet and characteristic, lymph node status, AGCC staging, oncogenomic, oncogenetic status, and therapy. And the facultative data set are metastatic localization and date, therapy response, and adverse event. And I am the coordinator of this cohort.
So our study is going to focus on metastatic melanoma patients receiving immunotherapy, either anti-PD-1 or anti-PD-1 combined with anti-CTLA-4 treatment. First, we're going to describe our population, compare it to randomized clinical trials, and then we're going to look at endpoints, 4 different endpoints: overall survival, progression-free survival, time to next treatment, and time to treatment discontinuation. We're going to look at their correlation, especially to the overall survival, and then try to compare it to what we can see in randomized clinical trials. Also, we're going to compare what happens to patients when they receive immunotherapy as a first line compared to as a later line. Among the 27,000 patients from the cohort, we selected 1,534 patients that were metastatic melanoma patients. Ambiguous patients were excluded, but we tried to keep as many as possible. So what we see here on the left is the patient described with major characteristics that are of interest for melanoma, among others. While performing this analysis, we saw a few things. First, that the protocol as defined for the Pilot 1 study is heavily focused on non-small cell lung cancer. And there are variables that are not present that would make a lot of sense for melanoma, such as phototype, a risk factor, or LDH, a prognosis factor, that are not present in the protocol. Some variables in the protocol doesn't really make much sense for melanoma on the other side, such as smoking status or EGFR status. We also see variables of common interest between melanoma and non-small cell lung cancer, such as breast flow thickness or histological type. And additional to the variables, there are, as any real-world data analysis, troubles with missing information and what to do about it. It impacts both variables of interest as well as definition for endpoints. And the framework is quite high level and flexible regarding those aspects. But having specification for those aspects would be important so as to reduce idiosyncratic choices that could lead to biases and hindering the reuse of the study, or hindering as well the comparability of this study with other studies. Specification is important, but on the other side, we have to make sure that it is not too constrained so as to maximize real-world evidence potential. And what is quite interesting as well is that although there was a common CRF between all the centers, there's still quite some heterogeneity in how the data was collected. So harmonization was still necessary. If we compare our population to randomized controlled trials population, we see that there are really few variables on which to compare them, namely here age and sex. And that goes to show that it is quite difficult to compare them, especially considering that randomized clinical trials are specialized populations. For example, there are generally ECOG 0 or 1, so really healthy individuals compared to real world. What we can see here on the age that is reported is that our real world data cohort is quite older, given its proportion of people above 75 years of age. What we can see for the endpoints here reported on the left are the overall survival, progression-free survival, the time to treatment discontinuation, and time to next treatment, is that first of all, they're quite well correlated to OS with the chart on the bottom right. They're all above 0.8, showing quite good Sugar-C potential for those endpoints. Progression was actually not that easy to calculate, and we had to resort to many different idiosyncratic choices. But the resulting progression-free survival seemed to be quite close to what we can see in clinical trials. Here on the left, we have a chart for this PFS. So in red, we have the real-world data, and in blue, we have randomized clinical trials, and we see that the values fall into a range of clinical trials showing consistency. On the right side of the chart, we have overall survival, and we see it's quite empty, because in clinical trials, overall survival is not usually reported, needing additional follow-up. So what we see here is that in terms of endpoints, it's quite difficult to compare clinical trials to real world, and PFS is really the bridge between the two. But we have the difficulty that PFS is kind of difficult to define and collect in the real world. What is quite nice for patients is that we see that the immunotherapies are quite successful in the real world, showing good translation from experiments to the real world.
So, first thing, no multicentric melanomas today on comparison between clinical trial and real world data has been performed until now in the field of melanoma. Second important point, we have analyzed patients which are often excluded from research clinical trial. And thus, we were able to demonstrate that older patients that generally are not included in the research clinical trial can have excellent results with immunotherapy, with the same efficiency than younger patients. And the second very important information is that checkpoint inhibitor are still of interest with good clinical response in later lines after 2 or 3 previous lines of treatment.
Some additional learnings and recommendations that we have learned from performing this study is that the framework is currently quite geared toward non-small cell lung cancer, and it would need some additions, extensions, improvements in order to be a framework for all of oncology. Second thing is that any adaptation and implementation that we needed to bring in order to deploy the framework to our study could be idiosyncratic. However, specifying too much, any choices could be too constrained, and although valid, could reduce the scope of the framework and real-world potential in general. We see that a common scheme on the data is that only what happens at the hospital is collected in data, and things such as progression is not really accurately captured by the data because it happens all along the patient's life. And another interesting fact is that data owners here at the RICMEL database were quite eager to participate to this study because they actually wanted to know the results of such a general analysis of population. Usually they do very specific analysis. Thank you very much.
Thank you, Adam, Maxime, and Brigitte for those wonderful presentations. I'd like to ask you to join me back on the screen. And I'd also like to invite Jennifer Christian, who's Vice President of Clinical Evidence and Epidemiology from IQVIA, for a follow-up discussion of these presentations and to further discuss what it means to broaden the application of a common framework to evaluate real-world endpoints. My name is Mark Stewart, and I'm Vice President of Science Policy at Friends of Cancer Research, and I've had the fortunate opportunity to partner with Laura Lassiter at Friends on our real-world evidence work over the past few years. And as what's been described over the course of these discussions today is that this has been a stepwise approach for us in terms of analyzing various real-world endpoints across different US data sources to arrive where we are today. And taking a step back to our pilot 1.0, what we were able to note is that we were able to use a common framework to extract similar populations and also noted some correlations between real-world endpoints like time to treatment discontinuation, time to next treatment with overall survival. But there were some noted limitations around these data sources that could make one pause a bit in terms of what you're able to learn from those efforts. And I'd like to start with Jenny, who's been also a part of this effort from the beginning, to kind of describe what some of those challenges were that we encountered in this initial work and why these subsequent analyses make sense and help complement some of those initial findings.
Thank you, Mark. So in the first pilot, we saw strikingly similar results in overall survival rates among frontline PD-L1 users across 6 different real-world data sets. And those findings were also comparable with findings from clinical trials that also assessed overall survival. But it did leave us with several questions. The first was how well still is survival captured in the real world data using these claims and EHRs, which I'll touch on in a moment. But another question was, would the results have been more aligned if we had restricted the population? So making it more homogeneous across the 6 data partners, but also in terms of aligning with a clinical trial. And so those results are underway now. And also, can we compare these endpoints across various treatment arms? Because 1.0 focused really on a single treatment arm of frontline PD-L1 inhibitors. And if we compared across treatment arms, would we expect to find similar results? So those were described in the first panel. And I highlight this because there have been a number of efforts that have come out of 1.0 that are still being explored. But for survival specifically, I think there's a lot of concern in the research community around missing death due to loss to follow-up because patients may no longer be seeking care at a particular clinic, whether it's from maybe getting sicker, seeking palliative care, nursing home support, transferring care, moving to family. There are a number of real-world situations that may limit how we can follow up on these patients. So there is a need to be able to link to other national data sources that would capture mortality, like the National Death Index. But unfortunately, there are a lot of issues in accessing that data and to do so in a timely manner. But in countries such as England, there's a national system that captures nearly 100% of all deaths. And the work that Adam shared found remarkable similarities in overall survival results, similar to the other real-world data sources that examine the same question. So to me, this suggests that mortality is maybe captured better than we thought. I thought this effort was successful in further supporting and potentially validating those results from 1.0.
Thanks, Jenny. And another question we had was, obviously our initial work was focused on non-small cell lung cancer, but how might this framework be operationalized in another disease setting? And in this instance, Maxime and Bridget both described the application of this framework in melanoma. Maxime, can you expand a bit on how you applied this framework in melanoma, and specifically, were there specific elements that needed to be adapted to make it fit for using it in melanoma?
Yes, sure. Thank you, Mark. So obviously, melanoma is a different cancer, although the treatment here is the same, and the variables of interest will vary. But although they might vary, they mostly revolve around risk factors, prognosis factors, and exclusion and inclusion criteria that we have in studies in order to compare them. For example, smoking status is not really relevant for melanoma, nor is EGFR. But on the other side, things such as phototype is really an important risk factor for melanoma. We also saw in the framework, when we were adapting it to our study, that it had strong use of time margins in order to make sure that some events were occurring, and safety in this data curation using time margins. But in our case, in our database, margins using time were not really appropriate for choices, notably because in the US database, there were concerns about patients switching from one system to another. We have a mention about switching from one health plan to another one, which could not happen in our case. The RICMEL database having a really good coverage of all the melanoma patients. Something that was quite different also was around the progression subject. It is not recorded as is, as expected of real world data, but information about treatment ending in progression actually was. So we used that information as a good proxy in order to reach progression. Of course, it's not the RECIST criterion progression, it is more the clinician, physician assessed progression. But it was a good proxy, and the results show quite consistency with clinical trials, so it's quite encouraging. And also, although we have time to next treatment and time to treatment discontinuation as good candidates as real world endpoints, I think it is good to have endpoints that are relating to patient state, such as overall survival and progression-free survival, because time to next treatment and time to treatment discontinuations are related to treatment choices, and they might vary, although they are really correlated to what happens to the patient, but it's not an immediate measurement of what happens to the patient. So those were main points about adapting the framework to our study.
Thanks, Maxime. And Adam, I'm curious as well. Maxime had mentioned it, and you also mentioned adaptations you needed. And I'm curious how you balance this need for, or this desire to have some harmonization and standardization, how this data is being extracted from data sources so that there's not only confidence in the underlying data, but also consistency in the populations that are being extracted, balancing that with also the desire to have flexibility so you can capture the uniqueness of different data sources. How do you balance those and grapple with those two needs?
Well, Mark, it's definitely a challenge. I think from our standpoint, the biggest thing the biggest obstacle we had to overcome was around the differences in when PD-L1, PD-1 inhibitors were approved in the UK versus when they were approved in the US. Since they were approved 2 years earlier in the US than they were in the UK, we had to adapt our protocol to be able to allow enough follow-up time on these patients who were diagnosed and treated at a later point in time so that we could get an accurate reflection of what those outcomes for those patients looked like. Additionally, since the data capture is different in the UK than it is in the US in that it's a national data source, it takes a longer period of time for all that information to be collected and to be usable and for the data quality to be assured at various points in that data collection. So in addition to making sure we had enough follow-up time on the patients, we also had to include some additional inclusion-exclusion criteria to our patient population to make sure that they had all of the relevant variables that were required in order to complete the assessment of these patients. I think another point not to overlook is that there are restrictions on some of the data elements within the CAS data set versus potentially a proprietary source or a privatized source of data collection. So in the UK, for example, some PD-L1 inhibitors are funded conditionally through a program called the Cancer Drug Fund. It's a managed access program where patients are given access to drugs. However, payers like NICE within the UK are assessing the outcomes of these patients sort of in privacy before making determinations on whether to fully authorize these products for use in the general public. So any data on outcomes for those patients are restricted. So we did have to use a smaller, more refined subset of PD-L1 treated patients, which will obviously have an outcome on some of those results. So while we did have similar patient populations in a number of areas, there were some specific adaptations around the data capture, the amount of time we were able to follow up the patients, and who was in our final cohort that differed considerably and things we had to take into account.
Thanks. And Ginny had started some of this in her opening remarks, but because in the CAS system, mortality data is quite complete, can you add some just additional thoughts on how this analysis helps strengthen or complement findings from the initial FRIENDS results?
Yeah, sure, absolutely. So I think two other things on the CAS dataset which are helpful to mention is that, it's a national registry, but it acts as a closed data set, meaning that we don't lose a lot of patients to follow up in terms of moving from one health system to another or moving from one provider to another. So it does give us very good longitudinality and capture of patients. Obviously, it's a nationally representative source as well, so that even though there are some limitations in who we're including, we know that we're going to get a representative population and that the results, the endpoints, the outputs might not be skewed to a small subset of patients that are being treated with one type of pathway versus another pathway. And then obviously having that confirmed mortality information was critically important. So not only do we get complete mortality information in the date of death, at various junctures throughout the data set, we're also able to get confirmation that a patient is alive. So we're not relying on things like visits or visit dates or recorded treatments in the data set to make those affirmations. So from us, having that overall survival number, understanding that we had the complete mortality information gave us a more reliable benchmark for comparing to some of the surrogate endpoints. So I think when you compare the overall survival from the initial US data sources to some of the surrogate endpoints, there was, and I think Jenny alluded to this, there was always some ambiguity around how well captured the mortality information was, and therefore how well could we consider the reliability of those surrogate endpoints. And considering that we had similar real-world overall survival in the cancer analysis system as we did in a lot of the US data sources, it gave us more affirmation of what we saw in the US data sources, therefore allowing for a stronger reliability in comparison to some of those surrogate endpoints, especially in time to treatment discontinuation, where death was included as an endpoint as well as the treatment discontinuation, whereas in time to next treatment, patients were censored at death, so that removed some of that certainty.
Thanks. And I know we're quickly reaching the end of our time here, but I wanted to kind of transition over to Professor Drino for a moment. Not only are we trying to apply similar definitions across these data sets, but you also have to think about the clinical interpretation of what these endpoints are telling you. And I'm curious from your perspective when looking at time to next treatment or time to treatment discontinuation for melanoma patients, how you might act upon that information in terms of the care you provide to patients and how you're interpreting those when it comes to assessing treatment outcomes.
Thank you, Mark. That's effectively a very important question for us, because when you are with progression-free survival or overall survival, so you are many information concerning the patient, but not really in link with the drug. And the difference is that when you have the time to next treatment is of particular interest in immunotherapy. That means that you can determine the duration without treatment between the end of immunotherapy and the next treatment. And we know that with immunotherapy, so this period can be very long, but we don't have exactly this information. So I think it's really important to have it. The second thing is concerning the discontinuation of the treatment. So it's also for us a manner to have the information about exactly what is the duration of the treatment for the patient, how long he was under this treatment before stopping, and why it has been stopped, either for adverse event or for progression. And today we know that probably the duration of immunotherapy is very important concerning the development of adverse event after stopping treatment. And the risk of adverse event after stopping treatments appear more important in patients with a long duration of treatment. All this information can be obtained with this kind of endpoint that you are unable to interpret it with overall survival or relapse-free survival.
Thanks. And also in the presentations, it was noted that you're able to arrive at some complementary information that you might not get from a clinical trial. Could you expand on that a bit in terms of what additional information you're able to gain from these real-world analyses, and how might this inform maybe next steps in terms of what data you might want to capture in the RIC melanoma database that you have?
So I think that what's very important is in our database, we include patients which generally correspond to exclusion criteria. I mean, by example, patients with brain metastasis or ocular melanoma and mucosal melanoma. And thus we were able to demonstrate that patients, I mean all patients more than 70 years old, often excluded from this trial, have the same clinical results and same tolerance than younger patients. That means for us, we are able now to think that immunotherapy, even in a patient 80 years old, can be discussed, as we know, by example, with anti-PD-1 that we have generally mild adverse event. And the second thing, which also is very important, we know now, so through our research, that even if you use immunotherapy after 1 or 2, even 3 lines of treatment, you can have a clinical response. Never it has been demonstrated through a trial, and that also for us is very important to discuss with our patient. Just to illustrate 2 examples why we think that the real life is very important to study today.
Thanks, Brigitte. I guess my last question, we're at the end of our time here, Adam. How are you noticing kind of differences in how real-world data is being used in the US versus outside the US? And are there learnings that we can pull from either of those?
That's an excellent question. So more and more in France, but also at the European level, our agency require for data after the labeling of the drug. So by example, moreover, they want that we develop code, so they require from the companies that they have cohort of patients for the follow-up. And then after, it could be very important to discuss mainly the price of the drug. We know, by example, that immunotherapy is very expensive. So this kind of data in the real world is also a manner for our agency to discuss the real interest for our patients at long term, and then to also discuss with the company the price. It seems that clearly in the future, it will take more and more important, and probably will be another endpoint very important. And personally, I support this approach.
And Adam, just a quick comment, and then we'll wrap up.
I think similar to what Brigitte was saying, on top of using real-world data to demonstrate the clinical efficacy of novel therapies like checkpoint inhibitors, we spend a lot more time demonstrating the cost effectiveness of these therapies. So patients who are treated with immune checkpoint inhibitors, what does their healthcare resource utilization look like? How often are they being hospitalized, being treated for adverse events, and using that to better understand whether or not more patients should be receiving these medicines or not.
Thanks, Adam. Well, thank you so much for the presentations and this follow-up discussion. I'd like to hand it over to Jeff to wrap up the meeting. Thank you.
Okay, thanks very much, Mark, and thanks to all of our participants in today's meeting. This project has been a true collaboration, and we really appreciate all of you tuning in today to explore the results of these impactful projects with us. Please join us tomorrow for our second session, again at noon Eastern time, where we'll transition the discussion to focus more on the policy implications and opportunities from this work and for the future of real-world evidence more generally. We'll start with an exciting discussion between FDA Principal Deputy Commissioner Amy Abernathy and the new head of bio, the President and CEO, Michelle McMurray-Heath. And that will be followed by a panel of experts that you won't want to miss. Thanks very much for joining us today, and we'll see you tomorrow.